Thank you, Dr. Sadiq. We have to move to our next speaker. We're changing crops. Our next speaker is Dr. Delphine Fleury from the University of Adelaide, telling us about the genetic basis of wheat yield under dry, hot climates. Thank you, Delphine.
Is it working? No. Good morning, everyone. I'm going to stand up here because I'm too short for the stage. It's my pleasure to be back in India. I always enjoy the food and the fantastic people here. So today, it's my pleasure to talk about what we do at University of Adelaide, and more exactly what we have done at the Australian Centre for Plant Functional Genomics that is now part of University of Adelaide. So today I'm going to talk about the genetic basis of wheat yield under dry and hot climates. And more exactly, what do you learn from attempting positional cloning of yield QTL? So first, to set the scene, how is the wheat cropping area in Australia? So what we grow is spring wheat, and we grow it during the winter season. So it can look a bit confusing at first for people coming from from Europe or the States. The average yield is very low. It's 1.8 ton per hectare. The reason is that we have permanent droughts. And what we call drought year are really, really bad year where the harvest might be less than 1 ton per hectare. Nevertheless, it's the total area of production is very large. As you know, it's a large country. You can see the area of production that is wrong one, sorry. That is in the south, south of Australia, so from western to south and Victoria and up to New South Wales and south of Queensland. And it's 70% of the production is exported, so it's a significant commodity, and a bad year can lead to a loss of up to $350 million. So the threat number one when you talk with the Australian breeders is yield, yield and yield. A bit more about the climate itself. So what we have, as I said, we have winter rainfall, and at the beginning of the season with cool temperature, and towards booting and heading stage, you start to have less water and your first cycle of drought stress. At the same time, temperature are increasing, so we end up with a high VPD and what we call terminal cyclic drought with heat wave. Now I'm going to talk about our strategy in quantitative genetics. For about 12 years, we have accumulated data on 3 mapping population of bred wheat: Racket 75 kukri, Excalibur kukri, Drysdale and Gladius. They were briefly described by Boris yesterday. They are all modern Australian varieties. The idea here was to work with the breeding company and find what they have already that is good in their germplasm. It's good to note that Drysdale was actually selected based on the carbon isotope discrimination, which is a proxy for transpiration efficiency. So that's a particularly interesting variety to compare mechanism and to compare QTL with other varieties. So first thing we've done is multiple trial in 2 or 3 countries for one of our population. Our Indian colleagues also did several trials on Excalibur kukri. And overall, across the 3 populations, we have more than 50 field trials. And we have QTL for yield, of course, also the yield component and other important threats that interact, that are a component of yield and also drought tolerance. And this is an example of what we get when we do a multi-environment QTL analysis. This one is, was done on Excalibur kukri double diploid population using data from Australia, Mexico, and India all together. And here that's where we can compare the yield QTL that you see as peak along the chromosome with days to heading, so phenology. And that's what Roberto will call the elephant in the room. That's what we obtain most of the time. The most stable, strongest yield QTL in dry and hot environment are genes that are already known and are not really yield per se, but actually are phenology. So here you have an example in chromosome 5A with the vernalization 1A here. If we look at the large QTL that is here, it's the strongest one for yield that perfectly match QTL for days to heading, and this one is VRN3. So that's one of the first characteristic of the yield QTL in dry environment in wheat. These genes are already used in routine by the breeder. So what we're after is other kind of new QTL and new genes. And the second characteristic of yield QTL in Australian environment and elsewhere in dry environment is the effect. The effect is, the main effect across environment is very little. So you have between 1, 3, 5% of yield increase from one allele compared to the second one. And that makes it challenging with all the error and the environment effect we have to chase this kind of effect and getting consistent trial to track them. A second thing, if we have already talked several times about it, but I'm going to go more in details here on the QTL by E interaction. Here you have, this is the double haploid RAC875 kukri population. We have 23 field trials in Australia and Mexico. Here the trials were ranked from the lowest yield in average across the population and check line up to Roseworthy, which was 2010, quite a wet year, and so it's ranked up to 5 tons per hectare. So the clear color here will be the harshest environment. And what you can see is some QTL are kind of stable. They are stable in the sense that the allele effect goes in the same, it is the same, it goes in the same direction. So above the line, it's the positive effect comes from RAC875 here. However, all the time we can see that the effect is always smaller when the environment is really dry and hot. That makes it even harder to follow and to understand. And an example that was taken by Boris as well was the chromosome 3B, but it's not a rare case. It's the case of QTL that are unstable between trial and go actually in different direction. So for example, here on the chromosome 1B, you have a QTL that has a positive effect in some environment, and the other allele, so the kukri allele, will give here the yield advantage. And we have the same on another QTL on 1B, 2B, and 3B, etc. And you find them everywhere in the genome. And it doesn't follow the ranking that I was using just before, from the harsh environment to the most optimal one. So how do we manage to understand this kind of QTL? What comes next? So what comes next is actually what we have done, and we have a few papers with colleagues. Whenever we could, whenever the technology allows us to measure physiological threat on a large population, we have done it on the same material that was used for yield. So usually we do that with colleagues who are plant physiologists. So for example, when Boris did his postdoc with us, he worked with the Lemnatech platform on the Drysdale gladius population, and we found QTL for growth rate and for transpiration rate, etc. And some of them match yield component that were found on the same population. We also worked with our colleague at University of Louvain in Belgium, with Rémi Chopac and Walid Zadok, who used the RAC 875 Q3 population to identify QTL that control transpiration and rate and night transpiration. And same, using our new genetic map that are high density genetic map, we could also look at the collocation of QTL with yield. Another study we have done is it's the measuring stomata threat, stomata size, stomata density, and it's also work that has been done on RAC 75 Q3. And with the new technology we have now for genomics, where we can generate easily high density genetic map that give a little bit more accuracy and also a better resolution if the population are large enough to compare QTL location. Today, Abtamu is going to present his work on the chromosome 1b comparing with growth rate QTL and yield QTL. So I invite you to look at his poster 123 this afternoon. The next, so the other strategy we've used is instead of using the double haploid population that doesn't have the enough resolution in terms of recombination to continue positional cloning, we've used a large collection of recombinant inbred line that we have from the same crosses. And for this, we start to focus on specific QTL and screen to get as many recombinants from in the region of interest, and then we've set up small facility next to our building, and we can actually do mini plot. So it will be 2 rows of 8 plants, taking care of the border effect. It will be in the ground, so it's a natural condition. Because they are mini plot, we don't need a large seed increase, which is one of the issues when you do field trial. You can imagine that when you have large collection of 7,000 recombinant in breadline, it's hard to have a kilo for each. So that's what we use, and we have also run-out shelter, irrigation, weather station, and gypsum block to actually monitor soil water potential, as you can see on this example here. And now I'm going to take one example that is our pet project on chromosome 3b to illustrate how we use all these facility, the physiological study, to actually try to find a yield gene. So this is our start point. This is a QTL that was described in TAG by Julien Bonneau. And this QTL was found to increase yield, early vigor, and 1,000 grain weight. Well, talking makes me thirsty. So my colleague in Montpellier loved that slide, and they show it all the time. So they spoiled my presentation a little bit. So typically what was shown in this paper in TAG is that the QTL3b has a really strong interaction with the environment, that the RAC 875 allele is positive and has a strong effect on yield and the other threats in a Mexican environment, while in most South Australian environment it didn't. And in our platform on the campus, it was a Q3 allele that was positive. So then Boris actually described a bit his method yesterday. And today I'm just following up on what's the outcome what the results from his research, the use of his model when we apply it to QTL. So in this case, we study 48 double haploid lines that have recombination points in the QTL3b interval. And using the facility here with mini plot, irrigation system, etc., as I described, first we monitored carefully temperature and soil water tension. And 3 environmental variables was described. The heat, which means temperature, drought from the soil water potential measurement, and the combination between the two. Unlike traditional way at looking at environment, we are not just using a trial, an environment as a factor here. What happened is that we calculated, Boris calculated, the environment, we quantified the environment as variable on period of time along the crop cycle. And from this, we could actually separate the G by E with the underlying components. So in this case, it calculated what is the allele advantage when there is no stress. So that will be the constitutive effect. When there's a heat stress, a drought effect, and the combination of drought and heat. And here is the result for the 3B QTL. So the first observation is the negative sign for the intercept. So the intercept is where you don't have a stress. That will be the optimal condition. So all the negative effect means that the Q3 allele is increasing the threat. So here, the Q3 allele increased positively yield, and it's highly significant by 10.9% when the conditions are optimal. Overall, there's a strong interaction between the QTL effect and heat, drought, or the combination of the two. If we look at what happened under drought, there's the Q3 allele has a strong interaction, a strong effect under drought for individual seed weight and also the seed number per spike. While the RAC allele is positive under heat stress and also under the combination of drought and heat. So that will explain why we had this apparent instability of the QTL effect across trial. And going back to this trial in Australia and Mexico on the double haploid population, so what we can see here is the allele effect. You have the zero line is the switch between what is negative is come from Q3. What is positive is yield increase coming from bracket 75 allele. And as you can see, the Mexican environment, it's where the temperature at flowering time is higher, about 25, and where the, there's a mistake, I think, here, there's a mistake, I think, in the graph, sorry about that. And when it's cooler, it's actually Q3 allele that takes over. I'd like to make a little break here, because when we come to this realization, then we thought at the same time, it would be interesting to see what the allele effects as well in other climates and other germplasm. So early on, when we started, when Julien started to work on the 3B QTL, we sent our germplasm in our lab, and we sent it in France at Biogema, and also in Turkey to ICMET. And it took a few years to do this kind of work, of course. So they did generated by backcrossing or also with a single seed descent to the near isogenic line and tested in their own germplasm, in their own climate, what was happening. I got these results only a few days ago, so I don't have yet the full description. But what I have already is that the first year of field trial in Castelnaudary in south of France, they only did the non-irrigated, so rain-fed environment. The trials was dry, and overall they had -8% of mean yield due to the dry condition. And I guess I don't have the temperature pattern. That's a thing I need to look at with Sébastien. But usually, as we know, it's dry and hot at the same time. So he observed when he introgressed the 3B QTL from Racket 75, when they introgressed into Altigo, which is a winter wheat germplasm, they have 2.2 yield increase and 2.4 thousand kernel weight. And this was done on 9 families of meals, and this was consistent with 3 replication. In Turkey, they introgressed the RAC allele doing 3 back crossing in Tosun Bay, their local varieties, and they also observed 4.3% yield increase under rain-fed condition and 2.5 under irrigated conditions. So that's very encouraging. That's very encouraging. Now I want to look really at the temperature pattern and see how it fits, and if we can learn something even more on when the RAC allele can have an effect. At the same time that we were working on RAC 75 Kukri, we worked also on the Drysdale gladius population. And Lance found that there's also a yield QTL on the chromosome 3B, and the location exactly matched the one of Racket 75 Kukri. And from the Drysdale and gladius population, what we learned, because the trial were done in different sites in Australia, some sites were in New South Wales. And New South Wales, it's like in Mexico, you have also a dry and hot climate, but you have deep soil, unlike Southern Australian sites. So we decided to set another facility, a pretty cheap one, because we use a rubbish bin to create a deep soil. Because in our bird cage, the polytunnel facility that I was describing just before, we have only shallow soil. So using this deep soil and under the rain-out shelter with gypsum block and controlling irrigation the same way, we could actually mimic the condition that we have elsewhere. And first, we verified that's how it's working. So the first trial that Pauline did for her PhD showed that the Racket 75 allele increased spike length per spike, early vigor, seminal root, tiller number, etc. So the number of spikes, the grain weight, biomass. So pretty much anything that is biomass related and yield. It's pretty hard to do position cloning of any traits in wheat because of the size of the genome. Just as a picture for people who work on other crops, half a chromosome, so 1 chromosome arm of wheat is the equivalent is the size of the entire rice genome. So when you look for gene, when you try to sequence, it's difficult and it costs a lot of money. So we were doing our work with the ongoing building up of genomic resources in wheat. And the resources that we usually use is Chinese Spring reference sequence. And also we had a project in Australia where Bioplatform Australia funded the shotgun sequencing of 16 varieties, most of them being Australian. It was a low coverage, but it enabled us, by comparing with the reference sequence, to actually identify many SNP. And it became suddenly relatively easy to find new markers. So using these resources, you can see here in red the QTL3B interval. It was at the beginning, probably about a year ago, Pauline had 75 genes in this interval, still too big to really study anything. With the new release of the RefSec version 1 a couple of months ago, we now have 22 genes in this interval. And so she used the Willy Bean facility to keep screening more recombinants in the very small region. So last trial I won on a 44 reel that had recombination point along this interval. And she had significant effect for the same threat that I listed before. The strongest effect were from Racket 75 on the leaf size, leaf area, and also the spike length. So using the peak of significance, she could narrow down the QTL effect on the 7 genes region. Then she generated near-isogenic line from this big collection of reals, because when you have a recombinant braid line at a stage F4, you still have a lot of residual heterozygosity that you can use to self-cross and quickly generate a near-isogenic line. And you have an example here. You have 4 pairs. In blue, you will have Kukri allele. In yellow, it's Racket 75 allele. The gray parts are actually fixed. So you can easily compare with a fixed genetic background, the line 13 to 14 that carry Racket 75 allele, and compare with the sibling that carry Kukri allele. So using that, she did some RNA extraction, and we thought, well, just start simple, start with seedling, because it seems that the threat is on early vigor, is very strong already, so let's start early. So she did the RNA extraction on this gene, and she just simply goes one by one on the interval of 7 genes. She just checks where do we have a significant difference in qPCR result. And very interestingly, we found one gene that is, we call it gene 7, that has a strong expression in all the line in Kukri, but also in all the line carrying Kukri allele, versus the yellow that carry the Racket 75 allele. This gene is actually a member of the proteasome system. Proteasome system is a complex of different proteins that is involved in the degradation of other proteins. So they are known to be regulator of other pathways. So our hypothesis here is that the Kukri allele carry a regulator that will actually target another protein and will actually degrade that target. While in Kukri, the target is not degraded. That's all we can say at this stage. We don't have more information on the mechanism, but that was a very exciting start. And the other genes that are surrounding on the same here, they are actually, they don't show a difference of expression like this. Pauline also looked at what the effect of droughts. So this is from an RNA experiment that was done in glasshouse. So there's no heat, it's drought. And it was a cyclic drought where we withhold the water in the pot. And you can see here the same, the Kukri allele is more expressed than the RAC allele. And in the control, the allele is more expressed than under drought. Okay, so just to summarize, so what we found very important is to monitor particularly the phenology when you do any yield study on a population. We also monitor the environmental condition, because by quantifying them, we can understand better what trigger QTL expression. And what we found this way is that the QTL3B has a strong dependency on temperature. Now, what we are doing is we are still using the Chinese spring reference sequence, which is not best. There's always a possibility that there's another gene in the region that we haven't found, that is only present in Racket 75. So we're doing local sequencing assembly of the region. And we're also screening, okay, some text is missing. We are screening some TILing mutants for this gene of interest as well. I would like to finish, I have 1 minute. I would like to finish to talk about what is next, the new project that we are working on. So for all this time, we've been working on Australian modern germplasm. Now we are looking at another step, which is allelic diversity. And as I said, it's very difficult to study yield component or drought tolerance in general because of the phenology, difference in flowering time in plants. So for this, for genome-wide association study, using diversity panel that come from different countries worldwide. We actually work in pot. I know what people will say, yes, we know it's not yield, it's only single plant component. But what it allows us, it's actually to tag each single plant for anthesis and then impose the drought and heat stress at a very specific stage of development. So at least we can compare things together. The other way that we are using is we are building a nested association mapping population, where we cross a donor line from different continents with a gladius and a scout, which are two modern variety. And that's also another way, which is a kind of a mixed study between biparental population and genome-wide association study. So hopefully at the next Interdrought, I will have more new results to present. And here we have many collaborators. So the funding bodies who supported us for many years. Pauline, Penny, Melissa, Paul, and Ute's group and their group in bioinformatics and we have people at AGT, so the breeding company who participated in the project very early on with the Australian trial. Matthew Reynolds with his team in CIMIT. The team of Meerut University did the work in India. And and Boris, we did all the physiological study. Thank you.